Hello and welcome to Invertebrate Paleontology and Paleobotany at Utah State University. This is lecture 40, where we'll look at the diversification of angiosperms and how that led to our own evolution and the proliferation of mammals and other animals during the late Cretaceous and into the Cenozoic. Now, if we were to go back in history to the Jurassic period, when there weren't any flowering plants, and most of the plants were ferns and conifers, as well as equisetum, the horsetails, and cycads and things like that. And we look at the animals that were living at that time. This is the sort of dinosaur fauna from the Jurassic of Utah here. We have big, huge sauropod dinosaurs like Apatosaurus and Diplodocus and Camarasaurus and Brachiosaurus. We had other fairly large dinosaurs like Stegosaurus and Ankylosaurs, and we had carnivorous dinosaurs as well and smaller dinosaurs. But if we look at all of those dinosaurs living during the Jurassic and look at their teeth, we'll notice that none of these dinosaurs were really doing much chewing. Even the herbivores. So the herbivores weren't really doing much chewing. Their teeth were more for plucking vegetation. So here we're looking at Diplodocus and Camarasaurus. These are the big, giant sauropods, so some of the largest terrestrial animals that ever existed. And they didn't have teeth that were really specialized for chewing or mastication in their jaws. Even Stegosaurus had sort of a plucking the predentary bone there, so it was plucking and eating food, but didn't really have any grinding teeth. They were all very pointy teeth during this period of time, during the Jurassic. If we contrast those teeth with the teeth that we see in dinosaurs from the Cretaceous, we notice that the teeth in these Cretaceous dinosaurs, in particular the ceratopsian dinosaurs, this includes the group that includes Triceratops, as well as the ornithopods. This is the group that includes Lamrosaurus and Parasaurolophus. These are the big sort of duck-billed dinosaurs. And they had very unique dental batteries. These are batteries of teeth that were ever growing and being ever replaced that provided a sort of a cheese grinder surface for vegetation that they were processing. So this is very different. These are very specialized skulls and jaws and teeth of these dinosaurs that were specialized for chewing. And we see these dinosaurs appear in the fossil record during the Cretaceous period. During the Cretaceous period, when we see the flowering plants becoming more and more diverse and a larger part of the flora existing during that time period. So this is a wonderful chart here showing that great diversification of the flowering plants that we see at the end of the Cretaceous and going into the Cenozoic. And if we look in the early Cretaceous, when we first see some of these flower plants, they were a really small component of the flora. So they were between about 10 and 5% of the flora was angiosperms. As we enter into the Campanian and Maastrichtian periods, the last part of the Cretaceous, we see that we get up to 20 to 30% of the flora are angiosperms. And then as we enter into the Cenozoic, we really begin to see that the flowering plants are really becoming diverse. Today they account for around 90 to 100% of the plant life that you see around you, and depends on the environment and the habitat. And during the Cenozoic, there's this big proliferation of the flowering plants. And this had a major impact on the animals that were living and feeding on these plants. Now, one of the questions we can ask is, did this advancement of the angiosperms lead to the rise of mammals? And one of the interesting things we can do is compare the diversity of mammals with that of the angiosperms. And we see that mammals were not very particularly diverse during the Mesozoic, though there's quite a few fossils being discovered now. But there's a big burst of especially herbivore animals that appear after the KT boundary with the extinction of the dinosaurs. But much of this diversity might have been led by the diversification of the flowering plants, that there were new food sources that these mammals could exploit during the Cenozoic that helped them rise in diversity. If we look at many of the Jurassic mammals that were living at the time, they're tiny insectivores. So they were carnivoristic. They were eating insects or other animals. Very few of them have any specialization for herbivory, or eating or exploiting plants. So they're mostly eating insects. The one exception to this is the rise of a group called the Multituberculous, which are a Mesozoic group of mammals. They become most diverse during the late Cretaceous and early Paleocene, and much of that might be for their very specialized way of eating. So if we look at a multituberculate, they have these procumbent incisors on the upper part and the lower parts of their jaw. This is very similar to what we see in many rodent groups living today. And we see a lot of incisors like this in many mammals that existed during this period of time, during the late Cretaceous and Paleocene, probably facilitating the grabbing and grasping of seeds and nuts and eating them and masticating them very quickly. Behind that, there's this really large tooth in the lower jaw. This is the lower fourth premolar, and the premolar is huge. It's like a giant circular blade in the jaw. And this giant tooth was probably used for shearing off the outer part of tough seeds or fruits, and getting down to the endosperm that was very nutritious, or even the germ cell of the seed to process it and eat it. Behind that giant huge tooth are these multicusped teeth. That's where they get their name, multituberculates, is because they have these molar teeth that have all these little teeny cusps, these little bumps on the surface, and those are used to crush and grind a lot of this matter, mostly coming from seeds and fruits, and then masticate it and chew it. And the multituberculates were extremely successful. They survived through the KT boundary and are very diverse both at the late Cretaceous and early Paleocene. So many of these smaller mammals are insectivores, or the multituberculates were the food source for dinosaurs. And so after the extinction of the dinosaurs, we begin to see the rise of some of these mammals as they get bigger, especially some of the mammals that are specialized for eating plants. Now, there are two groups of plants that really were very prolific during the late Cretaceous and in the Paleocene and Eocene. The first group is the Fabaceae. This is includes groups like the acorns and oaks that we begin to see at this time. And then the other one is the walnut family, the Julandiaceae. These are a group of plants that become extremely common, particularly in the Paleocene. Many of the leaves have been attributed to the walnut family during this period of time. And so this provided a food source for many of these mammals to exploit. So really at the end of the Cretaceous and the beginning of the Cenozoic, we see that the angiosperms are really diversifying. There's plenty of food, of seeds and nuts that are found. These are some fossil walnuts from the Eocene. And all of these food sources had a very major impact on the types of animals that we saw beginning to appear during this period of time, during the early Cenozoic and the late Cretaceous. We also see some of the first fruits appearing during the late Cretaceous and going into the Cenozoic. As we get into the Cenozoic, many of these fruits became larger and became more specialized in exploiting mammals for seed dispersal. And so we begin to see that early on in the Cenozoic, as many of these mammals become specialized in eating some of these fruits and nuts that are now available to them. During the Eocene, we see the first rodents here in North America and the first primates, the true primates, appearing in the fossil record. So this is Parames, that's the earliest rodent in North America, and Northarctica, an early primate. So the group that we belong to, the primates, appear during this time, and they appear to be exploiting and utilizing this new food source, the angiosperms, the flowering plants. So without the flowering plants, our own order, the primates, would probably not exist. All right, so one of the things I want to talk a little bit about is another interesting feature about animals and what they were eating in terms of changes in the flora that we can detect in the fossil record. And it's a wonderful story about the origin of grazing horses. So all horses that live today graze on things like hay and grass. But in the fossil record, many of the early horses were browsing horses. So this is a very famous diagram here of the phylogeny of the horses, and we have a wonderful fossil record of horses here in North America. And horses can be divided up into two groups. horses that are grazers, so eating mostly grass, and horses that are browsers. And browsers are going to be eating shrubs and saplings. They'll be eating nuts and berries and fruit and things like that. So they're exploiting different types of the flora that existed at that time. Now, the way that we can categorize these horses, these fossil horses, or whether they are grazers or browsers, is looking at the anatomy of their teeth. So in grazing horses, like this Miocene horse here, we see that there is this like cheese grater surface on the occlusal surface of the molars. So there's ridges that are up in valleys, and so the food would be ground between these. These teeth are self-sharpening because they're rows of enamel and cementum and dentine, which are softer. So you have harder parts and softer parts. So as they are grinding their teeth together to chew grass, they're grinding down the surface, but they're always exposing new, harder edges of the enamel. Below that is our browsing horse. This is an example. This is Eohippus, and it's very different. The teeth look very, very different. They're not as big, but the other thing you notice is that they have these bumps. So they have small bumps on here, and they're really designed for crushing and some slight chewing in those little basins, but they're not grinding back and forth, so they don't have a complete flat surface. They're very, what they call bunodont. They're very tall little cusps there. And so this is a tooth that's more designed for eating things like soft, fleshy fruits, and also leaves that are pretty soft to chew. Now, one of the things that causes teeth to wear down, especially if you're a grazer, is phytoliths. Phytoliths are bits and pieces of silica, of glass, that are actually secreted by grass, different types of grass plants. They secrete these bits and pieces of silica in the grass itself, and this is to deter animals from eating the grass. So phytoliths are found in many types of grass, and if you ever try to chew on grass and chew on it, it's like chewing on glass, bits and pieces of sand. It's really, really hard. And so plants secrete the silica as a way to keep animals from eating them, and these phytoliths are one of the major reasons that we see a lot of the grinding and wearing on teeth of grazing animals. They also are consuming a fair amount of dirt as well, and sand as well when they're chewing on grasses, rather than plucking at fruits and things that are higher up off the ground as well. So the teeth of grazing animals and grazing horses are, undergoing a lot more wear than you see in sort of the little browsing horses. So one of the interesting hypotheses that were out there in the 1990s was that the origin of grazing, when we see grazing horses during that period of time during the Miocene, was reflecting an expansion of the grasses. So the expansion of the grasslands across North America. And so it's predicted that we would see the appearance of phytoliths in the fossil record. And phytoliths preserve really well in the fossil record because they're made of silica. So oftentimes looking at sediment and oftentimes even scraping off the edges of fossils, you actually find evidence of phytoliths. So the question was, did we see the first appearance of phytoliths during the Miocene period? And the horses had to adapt to the expansion of the grasslands at this period of time. And this is the thesis work of Caroline Stromberg, who worked on this particular question. The remarkable thing that she found was that phytoliths were found in sediments going all the way back to the Cretaceous. We have lots of phytoliths in the Oligocene prior to this major change in grazing of horses. So what's interesting about this is that there was grasses throughout most of the Cenozoic, and grasses split off from the Angiosperms very early in this diversification, and there was grasses, but they weren't exploited as a food source until we get to the Miocene. And the reason for that might be that it was very difficult to eat. It was suboptimal food, and as long as there was other types of food out there, the horses would exploit that food. And when we get to the Miocene, several of the horses had adapted to being able to chew this food that had nasty little glass shards in it, which is very tough stuff. So today's all the horses that we have are all grazing, and they have this adaptation for chewing these grass with phytoliths in it. Now, another really cool thing that we can take a look at is a difference between two types of plants that do photosynthesis slightly different. These are what are called the C3 plants and the C4 plants. And they leave behind a chemical marker that we can study to understand how diets of various animals have changed in the geological past. C3 plants are called C3 because the carbon dioxide is first incorporated into a 3-carbon compound. The stoma, the openings in the leaves, are open throughout the day, and photosynthesis takes place throughout the leaf. It's more efficient with plants that are under cool and moist conditions and under normal light because it requires less machinery. Most plants are C3 plants. Now, C4 plants are called C4 because the carbon dioxide is first incorporated into a 4-carbon compound. The stoma are also open during the day, but photosynthesis takes place in the inner cells, and it requires a very specialized anatomy called Kratz anatomy. Now, C4 plants can photosynthesize faster than C3 plants, especially in conditions where there's high light intensity, so very shiny, very hot areas with hot temperatures. The C4 plants seem to thrive. They also have a better use of water, so they're more efficient in water use, and that's because they don't necessarily need to keep the stoma as open as much because they can photosynthesize faster. Now, C4 plants include thousands of plant species, at least 19 plant species. Many of the common examples include many grasses, corn, sugarcane, many of the summer annual plants, a lot of those plants that you can grow in very hot and dry places with full sun. Now, about 0.4% of the angiosperms are C4 plants, so they're a very small component of all the angiosperms that are out there. But they account for about 18% of the productivity of the plants out there, so of biomass. So quite a few of the C4 plants do quite well. This includes things like corn, sugarcane, very abundant plants today. Now, there's another type of plant. Those are the cam plants. Cam plants are kind of very specialized for very arid environments, and this includes many of the cactus and succulent plants that you're familiar with. And what these plants do is basically try to be as efficient in terms of water use as possible. The carbon dioxide is actually stored in form of an acid before it's used in photosynthesis. The stoma only open up at night, so they're very limited in the amount of time that the stoma open up for gas exchange. And this is to conserve water in very dry environments. They're very good at water efficiency, and that's because of the stoma. Now, when conditions are actually extremely arid, cam plants have the ability just to close off the stoma completely to conserve water, and then they actually sort of recycle the carbon dioxide in the cells inside the cactus. So they can do very, very effective way of conserving water in very dry environments. So most of these are very specialized succulents like cactuses, and there's actually not very many animals that eat these. So in C3 plants, they take in more carbon-12. So there's two stable isotopes of carbon, carbon-12 and carbon-13. And C3 plants take up have a higher frequency of carbon-12 to carbon-13. C4 plants have more carbon-13, and so they have a different ratio of carbon-13 to carbon-14, and it's enriched in carbon-13. So what's interesting about this is that we can trace the signal of the ratios of the two stable isotopes of carbon, carbon-12 and carbon-13, throughout the food chain, and we can do this in the past as well. So let's take an example. Let's say you have a goat, and this goat, you first start out, you feed C3 plants. So C3 plants would include things like apple, barley, grapes, oranges, potatoes, sugar beets, wheat. And you're feeding a goat this food, and the goat's doing well. If you take some hair, or you take a sample of bone or a sample of the teeth of an animal fed those types of plants, you would see that the ratio of carbon-12 to carbon-13 would be very low, so very little carbon-13 compared to the number of carbon-12. So something in the order of -24 to 22 based on a certain standard. Then if you switch the diet and you start feeding this goat corn and sugarcane and C4 plants, you would then see in the hair or the tissue of that goat, like the bones and the teeth, switch in that diet. You'd see a switch in the ratios of carbon-12 to carbon-13 over a short period of time. This is like in about 50 to 100 days, you'd see a complete change in the tissue depending on how fast that tissue grows. So we can actually then look at this in the fossil record to see how various animals switched and if they started to switch over and start eating C4 plants versus C3 plants and reconstruct the diet of some of these ancient creatures. Now I just put this quick graph up here to show that there is complete separation between the ratios that you see in C4 plants and C3 plants. So you grind up all of these and count up the isotope ratios of each one. They're very separate, and this is very useful. This chart is actually kind of nice because it threw in algae. So algae has a little bit larger range of the isotopes of carbon-12 and carbon-13, a little bit more overlap. But one way to rule out that they are not eating algae is to look at the amount of nitrogen in these samples. And so algae has very low amounts of nitrogen. So that's another way to, if you ever suspect that they might have been eating algae, though not very many terrestrial animals do, that's another thing to look at. Now, one of the really neat things you can do with this is look at the origin of cultivated plants, looking at the origin of agriculture. So this is looking at carbon isotopes from fossil human remains or prehistoric human remains from the eastern part of North America, going all the way back about 4,000 years ago or so. And one of the remarkable things that you can see is the advent of agriculture in terms of the use of corn. So the introduction of corn from Mexico into eastern North America about 1,000 years ago, so around 1000 AD, we see the diets of humans living in present-day New York and those areas suddenly starting to use agriculture or raise corn and starting to grind corn and make cornmeal as a primary part of their diet. And you see the carbon isotopes change to reflect this change. And so it's very useful for determining when species or when groups of animals change their diet with the introduction of new food. And we see that in ourselves and humans as we change to adapt to utilize agriculture that we see here in North America. So thank you again for watching this lecture video. If you're interested in taking a geology course at Utah State University, check out our website at geology.usu.edu. And if you're interested in who I am and the research that I do, check out my website at benjamin-berger.org. Thanks for watching.